going, just in the interest of time. And for those that join us a little bit late, that's totally fine. As we said, this will be recorded. But thank you everyone again for joining us. Our goal really today is to take a little bit of a step back and kind of take a big picture look at some basic features of feeder-free pluripotent stem cell culture. As we've always said before, please feel free to chime in with any questions. You can blurt them out, you can pop them in the chat, or feel free always to send us an email anytime afterwards as well. That's all A-OK. Specifically, as far as the agenda goes today, I'm going to do, as I said, a quick overview of some of the content that we've covered so far. I'll touch just very quickly on how we define stem cells and things like disease model generation, especially in the context of kind of the big picture of the stem cell and disease modeling workflow. We're then going to switch gears a bit and touch on a couple of different cell culture formats, right? Adherent, suspension, 3D organoid culture, just to name a few. And I'll name a few important considerations surrounding each. I'm then going to segue into PSC culture systems, as you see down here in number 3. And we're going to talk about things like feeder-dependent and feeder-free systems. We'll talk about things like matrices and substrates, media system considerations, and I'll give a brief introduction to passaging methods as well, which we'll sort of build on from here on out over the course of the next few lectures. We'll also talk a little bit about some challenges surrounding stem cell culture. If you want, we can also have one or two of you share some of your experiences or challenges in the lab. I'm sure we all have them. We all have some crazy stories from our work in this area, I'm sure. And of course, how you've managed to overcome some of those. And then at the end, as I always like to do, I'm going to close out today's session with a brief case study. This one's quite interesting. It's surrounding iPSCs in the context of immunology. Okay. So again, one more thing actually I forgot about this slide. Before the quick review, I thought this was a really cool picture that I stumbled across. Anyone know or want to take a guess at what these are? You can just pop them in the chat. And this is not a picture of Omar waking up in the morning either. Sorry, Omar, I'm teasing you a lot today.
I was gonna say sometimes it is, depending on the day.
Okay, so these are neural rosettes. Neural rosettes are one of the primary developmental signatures of neural progenitors in culture. And these particular ones were derived from human embryonic stem cells. You can see they kind of have this radial configuration, which is typical. So they diverge out right from a common center of these columnar cells in the middle here. And what we're kind of seeing here, which I think is super cool, is this maturation process kind of unfolding, in this particular case, using a marker called PAC6. PAC6 is something that's super important in the eye. In fact, it works in collaboration with another marker called SOX2, which we know is a useful marker for embryonic stem cells and neural stem cells. Pretty cool and pretty crazy image, though. One I thought would be just interesting to share. So last week, Omar took us down a reprogramming journey, if you will, one in which we sort of laid the foundation and touched on the advent of iPSC technology, specifically in the context of somatic cell reprogramming to generate our beloved induced pluripotent stem cells, or iPSCs, right? But again, it's always important, I think, to remember big picture stuff, and in this case, the definition of stem cells, which in the most basic sense abide by two primary rules, right? First, they can self-replicate, really as a means to make more of themselves, right? And then second, these are cells that can differentiate, importantly, into other cell types as well. And there's a lot of different types of stem cells, which we talked about in the first lecture especially, right? But they have essentially, a lot of them anyway, an unlimited capacity for self-renewal, like these pluripotent stem cells and embryonic stem cells, right? Embryonic stem cells derived from, say, a 5-day-old blastocyst, and then our iPSCs that, as Omar talked about, are generated from reprogrammed somatic cells. ESCs and iPSCs can become just about any cell type. And these are cells or even cell lines derived from undifferentiated progenitor cells that, again, are capable of forming multiple types of tissue after inactivation or activation of certain key genes. But we also talked a little bit as well about cells with a little bit more of a limited renewal capacity, right? These adult multipotent stem cells, for instance, on the right here. And again, just a couple of important distinctions on multipotent stem cells are that these cells can renew themselves, but they can only renew themselves a number of times, right? By no means indefinitely. And they only differentiate into certain cell types, right? Essentially the tissue from which they're isolated, as you can see here in this table on the right. Now, as we've alluded to, I'd say over the past couple of talks, we're capable of doing things like taking blood or skin cells, for instance, right? And then forcing the expression of certain protein transcription factors to reprogram those cells into iPSCs. Again, we can edit those cells to introduce mutations. We'll be talking about gene editing in a couple of lectures from now. And then we can differentiate those cells into a whole bunch of different highly functional cell types, right? Think cardiomyocytes, neurons, immune cells, et cetera, et cetera. The list goes on, right? And then from all that, generate really, really cool models and a host of downstream different applications. Again, these are just some important considerations on the right here in actually obtaining cells for iPSC generation. We always have to think about things like patient comfort, sample size, the ability or experience that you might need to reprogram a certain cell type, the method that you're using, the cost of the application, right? And always, of course, I think big picture, the intended downstream application is important to consider as well. And although, and I know you've seen a rendition of this slide before, Omar and I quite like to use this a lot, but I think it nicely puts things into context where we sort of highlight what the stem cell disease modeling workflow looks like from a high level, right? And how, importantly, all these individual components, right? Cell models, reprogramming, engineering, characterization, which can occur anywhere in the chain here, differentiation, and all different forms of analysis that we have at our fingertips really allow us to create these physiologically relevant models. And from that, then we can then study a whole host of different disorders. Again, we talked about this in the first lecture that I gave in the context of Parkinson's disease, right? And looking at iPSCs into dopaminergic neurons. But as I said, the case study I chose for today is a little bit different. It's one that looks at the use of engineered iPSCs for hypoimmunogenic studies, more specifically hypoimmunogenic derivatives of iPSCs that are actually able to evade immune rejection, which is pretty cool. And again, just to kind of drive that idea home here, there's a lot of promise in stem cells. We all work in a really, really cool and really rapidly advancing field. And I think stem cells are an important avenue into a lot of different things, right? We talked about basic biology, where we can learn to do things like prevent or treat birth defects or developmental processes. I did a lot of work in this arena before joining Thermo, looking at premature infants that were born with brain injury. Disease modeling and drug discovery, right? And this is a really cool one because we can use stem cells as a tool to then test the effects of experimental drugs, often on actual patient cells, right? In disease modeling, the sky's the limit. I think we're really just at the tip of the iceberg here in creating these patient-specific, right, or disease-relevant cell types to understand human disorders. And then last but not least, always there's cell therapy. There's so much promise in this, right? We're going to harness some of these inherited capabilities of stem cells and hopefully apply them to act as a repair kit, if you will. So lots of potential in the stem cell arena and a really good space to be in. So what I thought we could do now is just kick things off and segue into some of the basic science and a few different mechanisms underlying all this stem cell potential, right? And I'm going to touch on just a few of the merits of different types of cell culture formats that you might have encountered or you might yet to encounter along your journey in the stem cell arena. And I think, again, we'll start here by defining cell culture, right? And cell culture is really the process by which cells are grown under controlled conditions, right? Generally outside of their natural environment. After the cells of interest have been isolated from, say, a living tissue, right? Whether that's a human or a rodent, for example, or even reprogrammed into iPSCs, right? They can then subsequently be maintained under what should be nowadays anyway, quite carefully controlled parameters, right? Allowing us to optimize the workflow where we can use them as a means to an end, right? But not always the final product. So I think a nice analogy that I was taught here is to think of cell culture as a connecting flight, if you will, in that they typically aren't the final destination of our research, not always anyway, but rather a tool that we use kind of along the way to help study and improve upon all those different applications, some of which we just talked about. And along this journey, if you will, going back to our flight analogy here, you have to feed the cells regularly, right? You have to look at the cells routinely for important signs like their health, their morphology, right? You also have to subculture or passage the cells. And in doing so, we do things like remove spent medium. We transfer cells from a previous culture into, say, fresh growth medium. And again, all of this enables the further propagation of the cell line or the particular experiment that you're working on. And nowadays, of course, there's loads of different formats, right, that enable this process of cell culture, each of which are slightly different, you know, to each his own in what type of research you do and what the goals of your research are. But each of these has their own merits for sure, right? Adherent culture is quite standard nowadays. It's probably where most of your cell culture experience lies. There's suspension culture, right? We're seeing more and more of this nowadays as a means to generate really quite large numbers of cells that, you know, due to their shape and their size and other properties are more physiologically relevant than, say, traditional adherent culture. And then there's loads of different types of 3D organoid formats, right? Cardiomyocyte organoids, we've got neural organoids, et cetera, et cetera. And these are quite fascinating, right? And again, these more closely resemble in vivo cell environments. And we'll touch a little bit on each of these here in the next few slides. Now, just quickly here, I think this is a nice overview in that traditional cell culture systems, right? The adherent workflows that we've talked about mostly so far, what those do is they grow cells on these flat coated plastic dishes, as you know, in which the cells are dissociated, often enzymatically. And that works really, really well, as I said, for a lot of different applications. And it's appropriate for most cell types as well. And part of that is because adherent cells, anyway, nowadays, are quite easy to work with. And we also understand them quite well as a way to culture stem cells. But if you think about it, again, these cells and these systems are not necessarily representative of what occurs in the human body, right? They also, of course, require some form of tissue culture treated vessel, which allows us for things like cell adhesion and spreading to occur. Another important distinction, and I think something to think about here, is that growth in adherent cells is always limited by the surface area of the culture vessel that you're working with. What that means is it takes a lot more time and resources to, say, generate a large number of cells, right? Which unfortunately is what's needed nowadays for things like preclinical research, drug schemes, and a lot of different models, right? That for one reason or another require a lot of cells. And so nowadays, what we're seeing of, especially in say, Thermo Fisher, for instance, is cell lines that are adapted to spheroids in suspension culture. These are also nowadays quite easy to work with as well. We've got some really nice workflows that we'll share down the road in a later lecture, and some nice media systems that we employ here. They're easy to maintain. They need just a little bit of agitation to encourage things like gas exchange in vessels, in this case, that are not tissue culture treated. And suspension culture really gives us a nice, more predictive physiological response, should I say, than monolayer culture. And again, this is due to sort of this complex and multicellular nature of spheroids in suspension that really better mimics in vivo biology. And again, as I keep saying, allows us to generate a heck of a lot more cells, even in a short amount of time compared to, say, adherent culture. So in many ways, as I put over on the right here, but not always, suspension culture can be a nice bridge between, say, the research bench and the clinic, because it gives a closer resemblance to how organs fundamentally form and how they grow in the human body. So along those lines, again, this is a slide that you might see a few times from Omar and I, but I think this is a nice and interesting slide here because it helps put things into clinical context. And so what we're looking at here in this example is comparing expansion capability of suspension culture versus adherent culture workflows and highlighting a couple of differences in things like hands-on time involved, the cost of each workflow, potential pain points, etc., as well as importantly here, the capacity of each of these respective workflows to generate a large number of cells. In this case, 4.5 billion cells, right? And again, just to put this on a kind of a clinical context here, in preclinical research, this would be about the number of cells that you'd need to treat 2 patients with, say, a cardiomyocyte cell therapy. So in this slide here, you can see along the bottom that with traditional monolayer adherent culture, and starting on the left here with 1 well of a 6-well plate, it takes us about 28 days and 6 different culture vessel formats to basically scale up and generate our 4.5 billion cells. If we compare that, though, across the top row here, again, utilizing suspension culture here, it takes considerably less time, only about 19 days, as well as less culture formats to create the same number of cells. Think about this as well, that in addition to lowering the chance of contamination in suspension culture, because you reduce the hands-on time with less formats, right? Expanding PSCs in suspension uses a lot less media as well, oftentimes, to attain the same number of cells, often making it up to about 5 times less expensive. So, this isn't to say that adherent culture is bad. It's certainly not. Adherent culture is great. We've all used it loads and loads and loads, and we will continue to do so, I'm sure. But there are some benefits when utilizing suspension culture systems, again, especially if you're aiming to generate a large number of cells. And we'll expand upon this in a later lecture. And then finally, the last of the culture formats that I just wanted to briefly touch on is 3D culture. And yes, if we're sort of nuancing here, suspension culture is certainly a type of 3D cell culture, right? Especially when cells are allowed to aggregate and form spheroids or other 3D structures, right? Rather than being attached to, say, a surface in adherent culture. And we could spend a load of different time, right, talking about the different types of 3D culture models. But for the sake of today's talk, and because it's something that we'll talk about down the road as well, I did want to briefly mention organoids. And organoids are 3D structures that are typically derived from either pluripotent stem cells and or in some cases organ progenitors from, say, tissue-specific stem cells. in which cells spontaneously organize into differentiated functional cell types and progenitors. They also typically quite nicely resemble their in vivo counterpart, and they often mimic a lot of the functionality then of, say, a particular organ, hence the name, right? An organoid, right? So they're more complex. They're more in vivo-like than, say, even spheroids in suspension culture. And again, this gives us a nice microenvironment for things like targeted research, cell therapies, focused disease models. And as I said, we really won't go too much into these today, but I just want to kind of give a quick overview here, know that they exist, and really just kind of highlight them as a nice way, a nice diverse way to study different types of cell culture formats in a number of different applications. The other thing, just quickly, that's worth a mention here is cell culture vessel types, right? And again, there's loads of different types of those nowadays, right? As you can see here on this slide on the right. And again, we won't go into too much detail here in the interest of time. Also, don't want to bore you to death either, but some of these culture vessels I've listed here in the context of adherent versus suspension culture. And this is an important decision to make, right? You have to choose the right vessel format, which requires a good deal of planning in your experiment, both in the short and the long term, right? As I talked about with adherent versus suspension culture, if you're going to generate a large number of cells, you need to plan for that from a materials perspective as well. And you have to choose a vessel format that's designed rightly for your experiment, right? Thinking about things like culture consistency, the health of your cells, reproducibility, et cetera, et cetera. There's a lot of different types of different plastics, et cetera, that have been used over the years to cell culture and do cell-based assays, right? But I'd say probably the most frequently used one of these nowadays is polystyrene or PS. These are quite low cost. They have an inert chemistry, which makes them a good choice for disposable formats, right? They're chemically treated, usually with the treated culture surface typically carrying a positive charge, while proteins on, say, a cell membrane, for instance, are usually negative charged. This is why cells are easily attached to or attracted to cell culture grade plastics. You can also, and we'll talk about this a little bit later on, you can optimize these vessels by coating the surface, right? We use peptides, things like poly-D-lysine or PDL, different proteins like collagen, for instance, polysaccharides, right? And so there's lots of different shapes, forms, and sizes here and chemical configurations of cell culture vessels, right? Again, all just really things to keep in mind when planning an experiment. So I'll segue now into a quick discussion on a few more details surrounding cell culture systems. I want to touch on a couple of different essential elements of successfully growing pluripotent stem cells, and then more importantly as well, highlight some of the merits of feeder-free culture. So there's things that you have to consider, right? The matrix, the substrate, what the cell's attached to, right? And it's really important in facilitating the growth of the cells that you're working with. You have to think about the media system, right? And I would say maybe that the media system is maybe the most important component of the culture environment, right? Think about media systems that we use in the lab, right? They give necessary nutrients, they give growth factors, they give different hormones for cell growth. They help regulate things like pH, the osmotic pressure of the culture, right? We also have to think about the passaging method, right? That process of transferring your cells into a new dish, right? Which of course enables further propagation of the cell line to ensure that not only you're culturing enough cells for experiments, but also you're maintaining optimal health of your precious cells that you're working with. And I'm sure you've heard some of these terms before, right? But these are a couple of things that we'll throw around in some of our discussions on cell culture that I think are just important to define. We often talk about subculturing or passaging, splitting of the cells, right? As I mentioned on the last slide, this is really important. It allows us to kind of housekeep, if you will, on a routine basis to help divide and transfer the cells into a new culture vessel, to give them enough space and all the necessary nutrients, right, to continue to grow and thrive. Another important term is confluency, right? I'm sure most of you are familiar with this, but this is one of the primary measures of when to actually perform this subculturing task, which is something that you'll do quite regularly. And again, as I'm sure many of you know, confluency is an estimate of the percentage of culture vessel surface that's covered by your cells when observed by, say, microscopy. For example, 50% confluency means that half of the surface of the culture disc or floss is covered in cells. One thing just to mention here, and again, we'll talk about this down the road, is when we talk about suspension culture,
If you have read, then just mention, give your signature there.
we use in suspension culture, we use slightly different terminology here. Oftentimes, instead of confluency, we use the term density to describe the number of cells per milliliter. So this is a quick exercise here looking at estimating confluency. Anyone have an idea? And you can just put these in the chat, what the confluency would be in, say, box 1.
80%.
Very good.
Isn't it too high?
Yeah, I'd say about 80 to 90% time to passage those cells, right? How about in 2?
Closer to, we have 60 in the
Yeah,
55.
yeah, I'd say 50 to 60.
Yeah.
A little bit less here, maybe 30 to 40% confluency. And then maybe in 4, we're down to, I don't know, 10 to 15% confluency, perhaps, right? So again, this is just a nice, I don't want to say quick and dirty, but it is a nice quick and dirty way to estimate confluency and just kind of checking in on your cells, seeing how they look, seeing how crowded they are, because things like this really do impact their health and, of course, their viability. Now, a bit more terminology here. We're also really interested, for obvious reasons, in the number of cells per given volume that are actually alive, right? In other words, the number of viable cells. This typically relies on a device called a hemocytometer, right? A hemocytometer is just a small glass chamber. It kind of resembles, I've always thought of it, looks like a thick microscope slide, right? And we use this to determine the number of viable cells. And this is typically given as a percentage of the number of cells per volume. Most, I would say, protocols, all of ours do anyway, usually give you kind of a target viability range at certain crossroads in the workflow, right? It's a nice handy point of reference to make sure you're on the right track. And as well, I'll also include a couple of sample calculation slides in the deck that I share with you guys afterwards, just as a nice point of reference, if you will. Another important piece of terminology is colonies, right? Stem cells are almost always cultured in colonies, which are clusters of cells growing closely together in a dish. Omar talks about this all the time, but stem cells like to be together, and this close contact helps them to survive, right? They want to be in contact with each other. And then lastly, down here at the bottom, we also talk about plaques sometimes. These are essentially holes in a layer of cells. They can often be caused by things like a virus, and they're often signifying things like cell death or lysis or kind of apoptotic areas within the colony. What can happen in some cases is the virus, for instance, when replicate, it spreads and then it generates this region of cell destruction or lysis that are visible to the naked eye. So now that we have a couple of different terms under our belt, I did want to talk again about a couple of essential components that we need to grow PSCs. I've listed some of these on the slide here, but again, keep in mind that successful culturing of PSCs really requires an optimal combination of these different elements, things like media, matrices, and passaging that are all really in sync with one another, systems that work well with each other, right, to help best support things like, again, your cell health and your pluripotency. So when human cells were first, oh, go ahead, Omar. Yeah.
Sorry, just wanted to mention one other quick thing on the last slide. So,
Yeah, please.
I want folks to just the reason why we're harping so much on the terminology and the basics of the culture here is specifically for stem cells. We discussed this a little bit in the past 2 lectures, right? But these are transient populations of cells, meaning that normally in the embryo, right, these pluripotent stem cells that are found in the inner cell mass typically differentiate and then they give rise to the rest of the embryo proper. So these cells do not persist in the adult body or the growing fetus, right? And in our culture conditions, if we really wanted to maintain these cells as pluripotent stem cells, right, maintain them in that state that they typically don't stay a very long time in, we have to make sure that we provide them with the right culture conditions. And in that sense, the reason why PSC specifically are it's important to pay attention to their culture is because of their capacity to change, right? If you do not keep these cells in culture with the right conditions, they may still be alive, but then they can start to transition out of that pluripotent state, right? And you don't want that up until you specifically want to start differentiating your cells. But that's why these things that Tom is pointing to are very, very important considerations in basic PSC culture.
Absolutely. Thanks, Omar. Good point to call out for sure. So kind of from a historical perspective here, when human ESCs were derived first and foremost by Jamie Thompson in the late 90s or so, 1998, I believe it was, what we had for a system employed in those days were cells cultured on mitotically inactivated MEFs, right? Mouse embryonic fibroblasts, typically with medium supplemented with things like hPS or fetal bovine serum. And these fibroblasts or feeder cells, what they did is they provide growth. I shouldn't use them in the past tense because people still use them quite a lot today, but feeder cells or fibroblasts, right, in this case, provide growth factors and different extracellular matrix proteins, but they're not actually dividing themselves. And what that enables them to do is they secrete things like different growth factors and cytokines, which of course help enable crucial cellular functions, things like pluripotency and proliferation, and serve really as a nice support system or network for the pluripotent stem cells to grow on top of. This was super important pioneering work, I would say, that laid really an important foundation for the development of a lot of PSC culture systems that we use today. Now, to reduce the risk of contamination from proteins derived from all those different organisms and other adventitious agents, these MEFs in these feeder-dependent cultures are often replaced nowadays with human-derived alternatives, right? Things like human foreskin fibroblasts, right? They also eventually replaced or often replace things like FBS with, say, a knockout serum replacement. And again, this is always done, and these types of things are done to reduce the use of not only animal components, right, but also cut down on things like variability. I would also say, just thinking big picture here, a lot of the evolution of PSC culture systems, the interest for that is often driven by generating stem cells suitable for clinical application, right? We're selfish, right? We want things that we can use for ourselves to make our condition better, right? So what we're seeing a lot more of nowadays is things that are devoid of non-human animal material, right? Making them things like xeno-free, which again, I think makes sense in the grand scheme of things because we want to correlate the work that we do back to physiologically relevant human models. And so in the context of these feeder-dependent culture methods, it's always important to consider things like the preparation and quality of the feeder cells. There's a lot of things that can unfortunately go wrong in feeder culture, right? You can have contaminants like mycoplasmas, incomplete activation of the cell cycle can have the MEFs completely overgrow and go completely wild. They'll then outcompete the slower growing PSCs. And so when you're working with feeder dependent culture, right, it's really, really important to undergo proper maintenance of the feeder systems, which typically involves things like daily media changes. You have to check in on the cells, usually daily in feeder dependent culture, looking at things like morphology, cell health, confluency, as we talked about. What we do though with MEF feeder cells is they're typically mitotically inactivated before being used. This is often done treating them with things like mitomycin C or radiation. What that does is it arrests the cell cycle. And so really, I just wanted to touch quickly on feeder-free or feeder-dependent cultures here, feeder-dependent cultures, which rely on those feeder cells to provide a lot of other proteins, most often, again, growth factors and extracellular matrix proteins that we know are necessary for PSCs to grow in culture. And again, these are quite widely used today. Eventually, though, I would say there was a big trend towards media matrices developed to support PSCs completely independent of feeder cells, which at the time was a really incredible concept. And in feeder-free cultures, right, we're essentially working on the principle of omitting those feeder cells and then supplementing or developing the media itself, right, with all the necessary culture components. Again, put this in the media to compensate for things like nutrients, growth factors, even extracellular matrix components that would be otherwise missing. And so, again, coming full circle here back to about 2011 or so, we had some really nice contributions again from the Thompson lab, who basically established and published a fully defined feeder-free culture system that then allowed us to support the growth of PSCs without any animal components. And this is a product of ours at Thermo Fisher called Essential 8 Medium. Not all applications, though, require xeno-free, as we talked about before, or even animal origin-free media, right? A lot of the newer, more advanced PSC applications actually benefit from more complex mediums even, right, than, say, Essential 8, that are even more optimized to support things like challenging applications, think things like single-cell passaging and gene editing. And this led to the development of media systems like StemFlex. This is another media system that we have that's geared towards really, really good performance, maintenance of the stem cell qualities that I talked about, like pluripotency, and again, of course, survival in different advanced applications, despite all the stress that the PSCs are undergoing during things like challenging workflows. And again, we'll talk about a couple of different media considerations in a minute. I put a couple of pictures down here on the bottom of PSCs grown under feeder-free conditions. On the left here are human episomal hPSCs grown on Geltrex plates in Essential 8 media. And then on the right here, we have cells grown under the same conditions, although at 5x magnification instead of 10x. You can see here that part of the colony remains intact, while another part shows these kind of loosely arranged flatter cells, which is often indicative of differentiation. Now, if we consider again, as Omar alluded to a second ago, some of the necessary requirements for growing PSCs, growing or culturing cells on flat plasticware results in this sort of artificial two-dimensional sheet of cells, right? And, I think we've talked about this before, but the cells in the body experience much more of a three-dimensional environment, right? In which they're completely surrounded by other cells, right? Membranes, fibrous layers, adhesion proteins. So in adherent culture anyway, we need to provide cells with things like an extracellular matrix, right? Scaffold or a protein to help us more closely mimic our in vivo-like morphology and give these adherent cells a much more physiologically relevant microenvironment, right? One that also enables more realistic intracellular interactions as well. And so in feeder-dependent culture, we often rely on human or murine fibroblasts to serve as our matrix. And I put a nice table of some matrix considerations down at the bottom here. Selecting the appropriate matrix is really, really important. It will do you wonders if you pick the right matrix for your particular experiment. There's obviously a lot of choices. As I said, I've put some of these down in a table here at the bottom, like Geltrex, for instance. Geltrex is a soluble form of basement membrane extracted from murine tumors. We use Geltrex mostly for routine culture. It works really, really well. We also offer different variants of vitronectin, right, as you see here. Vitronectin is a xeno-free recombinant human protein. It was also designed in the lab of James Thompson. It works well with Essential 8. And this particular variant that I've listed here supports human PSC attachment and survival better than, say, a wild-type vitronectin. Geltrex and vitronectin are also compatible with things like single-cell passaging, although when we do that, we do typically suggest supplementing with something called Revitacell, which helps ensure attachment of single cells after passaging. And then lastly, at the bottom here, I've included laminin 521. This is another defined xeno-free medium that we know supports feeder-free culture quite well. Laminin is something that's present in blastocysts, and so it gives PSCs an environment that mimics the human stem cell niche. It's also really handy in that it provides good amount of further support for more challenging applications. Again, things like reprogramming, single cell passaging, clonal expansion, et cetera, et cetera. When you're selecting a matrix, one thing to always think about is following the protocol and those given guidelines, right? You have to consider things like the thawing temperature, the stability of that particular matrix, the diluents, right? These are all really, really important factors that help control the integrity of the matrix and therefore help control your cells, right? Matrices have typically an initial coating concentration, but they may need to be optimized as with anything for a particular cell line, and of course the intended downstream application. Typically, though, what we'll see is you'll dilute your matrix of choice. You use it to coat your culture vessel, often about 1 to 2 mils in a 6-well plate, for instance. We then incubate those matrix-coated plates, again at the recommended temperature and time, after which those coated cells can be used to culture PSCs. So really, the take home here is that the matrix is a really, really important determinant for successful culture in feeder-free conditions because it plays such a substantial role in things like normal cell growth. And if not done well, it can really influence factors like pluripotency and cell identity. Now, another really important workflow consideration, and I would say, again, perhaps the most important of these is media choice, right? There's loads of different media systems, especially those for different applications. A lot of different choices as well within certain companies. But I did want to touch on media just for a second here. And as I've alluded to before, there's been a lot of advances in media systems over time, again, all the way from, say, Jamie Thompson's contributions in the late 90s through to some big leaps and bounds, as I mentioned, in the 2000s, where we sort of had a re-examination or a relook at the media landscape, if you will. We tested newer combinations, often with, say, fewer components, and we defined more media, different media systems, right, to help support the growth of cells, giving rise to things like Essential 8, which sort of led to different media systems like E8 Flex and StemFlex, which you might have heard of, that nowadays not only support cells, but allow for things like weekend-free feeding through stabilization of factors like FGF, which I'm going to talk about in a minute, as an important component of some of our feeder-free media systems. This slide is a nice, because we talked about it a little bit, side-by-side comparison of a couple of the basic components that comprise feeder-dependent and feeder-free media systems. Again, feeder-dependent media culture systems generally support pluripotency and health using things like a DMEM-based medium supplemented with, say, fibroblast growth factor and or serum, or more commonly knockout serum replacement, which again is more defined and reliable serum-free alternative for PSC culture. The feeder-dependent cultures, again, as I talked about, do rely on feeder cells, which provide those different growth factors, extracellular matrix proteins, etc. But nowadays, what we are seeing a lot more of is feeder-free media, right, as a popular choice for researchers. Again, feeder-free cultures work on the principle of basically omitting those feeder cells and then supplementing the culture components to compensate for things like nutrients, growth factors, extracellular matrix proteins that are missing, right? And I've used an example here on the side of Essential 8, which again is a really defined media system that contains the 8 essential components necessary to support PSCs, hence the name, right? Essential 8. E8 is a really nice choice for researchers that need a defined formulation. Again, it has only the required components needed for culturing PSCs, which in the big scheme of things, right, amounts to a lot less variability than a lot of other media systems. And again, this is often the result of things like less interactions between those very limited 8 components and a more simple defined substrate, right? We also remove BSA. As you see down at the bottom here, BSA can be quite a potential culprit of variability. They have a nice chart down here at the bottom, how different batches of BSA in other media systems can actually lead to a lot of variability in their capacity to support proliferation of PSCs. And so with E8 especially, we see a lot less variability, especially when we pair it with, say, a defined matrix like vitronectin, for instance, even in more challenging workflow applications. I also want to just talk very quickly about changing the media, right? Routinely changing your media is an important consideration as far as the health of your cells. And a lot of workflows do require daily media changes, which is certainly fine. But some workflows nowadays are quite nice in that they allow you to skip a day here and there. So not quite as absolute as the slide implies here. Keep in mind, too, that we talked about this a little bit with suspension versus adherent culture. When you're handling the cells a lot, right, especially say with daily media changes, that does increase the likelihood of contamination, again, really by more hands-on time and potentially even an increased potential for user error. So it's nice to have support if you do have to do daily media changes. It's nice to work with a team. We've all been there. We've all come in on the weekend to change the media in our cells, I'm sure. But one thing to keep in mind here that I really wanted to call out is a lot of the need for daily media changes and a lot of, I'd say nowadays, the consideration that went into designing more flexible media systems is based around FGF stability. As you see at the top here, FGF is fibroblast growth factor. It's a potent regulator of processes like differentiation and proliferation in stem cells. And so in a lot of different ways, it really just, I would say, orchestrates the pluripotency process. FGF is also heat sensitive. Therefore, it unfortunately has quite poor stability at 37 degrees Celsius, meaning that we need to regularly feed our cells to help maintain their pluripotency. This is a nice figure here, I think, that shows the loss of or fluctuations in FGF activity, which can lead to, of course, inconsistent culture conditions. You can see here just how much FGF levels can dip on a daily basis. And so what our company, for instance, has done, and others have done this as well, is develop media formulations that eliminate the need for daily feeding, right, by extending the activity of FGF and other heat-sensitive components that we would typically see in PSC medium, which you can see here really limits these fluctuations, right? We cleverly call this media system E8 Flex or Essential 8 Flex, because as the name implies, it gives us a good deal of flexibility actually as to when you have to feed your cells. And so with E8 Flex, for example, media feeding can be skipped for up to about, I think it's 2 or 3 days in a week, including 2 consecutive days without compromising important things like pluripotency and genetic stability, which of course is really, really nice. And again,
Hey,
this
Tom.
slide's just, yeah, go for it, Omar.
I just wanted to kind of emphasize something here for folks in terms of media, but also the matrix. You'll notice that initially the goal was to get these cultures to work, right? In the late 90s, we started to get these cultures to work pretty decently, but there was a lot of variability. So we were trying to reduce the amount of variability by controlling the different components and things that the cells were exposed to, right? And to that point as well, if you look at this slide here that Tom's showing, right, when it comes to FGF2 activity, one thing that's very, very important that I would say to really always keep in mind is making sure that your cultures aren't going through the ups and downs, right? That there isn't a big change overall in your culture throughout the culture period. This will help maintain your cells pluripotent and carry typically normal. But if that's the goal, right, looking at those older media systems that Tom is showing, where the concentration of FGF drops dramatically within that first 24 hours, right? So you imagine you go into the lab and you feed your cells in the morning. Before you go back home, the FGF concentration in your media is now almost down to half, right? Which is a huge change over, the course of just 10 hours. Now that we know a little bit more about the media and then those different components in the that can, for example, stabilize FGF2 activity, we can tweak our formulations now to make sure that we address that issue and start to really make sure, again, that our cells maintain somewhat of a homeostatic condition across the entire culture period.
Super important. Thanks, Omar. One other thing that Omar kind of alluded to, and I think just to drive that notion home then, is things like media choice are super important considerations for the stem cell workflow. There's a lot of different choices, as I mentioned, even within the same company, that allow you to optimize basic components of your research, right? And account for things like, as you see on the slide here, scalability, robustness, flexible feeding schedule, right? Culture systems aspects or particular aspects or parts of the workflow. I'd say StemFlex in the middle here is probably our most well-rounded and versatile media. I've also not listed it here, but since we touched on it a little bit in the beginning, we are performing a lot of work nowadays in 3D culture as well, rather than 2D or flat culture. And so, in suspension-based systems or organoid systems, right, 3D culture formats, right, a good question here, and just to call this out, is why would a researcher consider suspension or 3D culture over, say, adherent or 2D culture? I heard someone say in a talk once a number of years ago at an ISSCR conference, that the world is not flat nor two-dimensional, right? So why should your cell culture be? And that always stuck with me because it's just I think, easy analogy to understand, but it's an important analogy when we're trying to make models that are relevant to the human condition, right? And so as we've talked about, more traditional culture systems, right, they grow cells on flat coated plastic dishes, but again, these systems are not always representative of what occurs in the body. And so a lot of cell lines can be adapted to things like spheroids and suspension culture or organoids, as you see here, which again, generally speaking, give us a more predictive physiological response due to that sort of multicellular and complex nature of cells in 3D that better mimics in vivo biology. And again, I had a slide on this a little while ago, but this is a nice bridge always between the research bench and the clinic. And so again, we'll talk about this when we talk a little bit more about 3D systems, right? But we have a nice media system called StemScale, which was designed to support large-scale generation of PSCs in 3D. And we'll go into that workflow and some of its applications, as I said, in a later discussion. Then lastly here, kind of along the lines of bench to bedside, this is just a slide I found in one of our older decks highlighting an overview of 3D models in the setting of drug development, just to put things into context of a typical workflow of a drug going from bench to patient, right? And I think, again, highlights the utility of 2D and 3D models in the translational setting. We start on the left here with, say, a screen of loads of different compounds in, say, I don't know, a plate-based assay, where we can find positive hits. We can test them in 2D monolayer to see the effects in a living system, quote unquote. And basically, by testing in 2D systems, when we're doing things like drug development, what that does is enables the researcher to fail earlier on and much more cheaply, rather than venturing into more expensive or more complicated 3D models or even subsequent clinical trials, right? And again, all this testing that we do early on allows us to evaluate things like efficacy, mechanism of action, functionality, toxicity, and so on and so forth, right? We then take that data and we use those to further test different compounds in animal and eventually human studies, right? Which, again, as I said, typically require a significant increase in cost and time. And so, all these different types of models are, again, a nice bridge between, say, lead discovery and in vivo studies. And not only do they recapitulate the human state in a more physiologically relevant manner, but they allow us to reduce some of the costs early on anyway, often associated with this drug discovery. And I can't remember the exact cost of what it takes to bring a drug to market. I'm not sure, Omar, if you remember the exact number, but it's massive. It's absolutely massive. And it takes a lot of time and energy just to get it to that state. One
I
other
think
thing that, go ahead, Omar.
it's in the billions. I think it's
Yeah,
about $10
it's
billion
crazy.
to put one drug on the market. Yeah.
Yeah, which is absolutely wild. And so again, just speaking, I don't want to badmouth adherent or 2D culture because it's certainly very useful. One other thing that we will talk about later on in the course as well that I just thought of is automation. Automation nowadays is very much finding its footing or presence in a lot of the workflows that we do, especially at larger scales. And when we talk about automation, we'll be thinking about things like how the use of different machines and different software, for instance, right, can essentially replace, or I guess a better term would maybe augment some of the manual processes that we're doing, thinking about things that we're talking about today, like feeding, passaging, media exchanges, for instance, to ultimately help accelerate and cheapen processes like this, right, and lead to more efficient, reproducible, and even scalability within the workflows that you're working with. But I digress anyway. But this is a nice big picture representation, and I think on the whole, a useful concept to ponder on. All of that said, we often, of course, have to consider the cells themselves, right? You have to have stem cells, otherwise none of this is possible, right? And again, we won't go into too much detail here. This is just an intro to feeder-free culture, but these are some basic characteristics of what you want your cells to have in culture that can be telltale signs of happy and healthy cells. The picture on the left here is essentially in vitro nectin, a combination that I mentioned earlier, and you can see how nicely and tightly packed these colonies are. They've got nice, defined, refractive-looking edges. Omar mentioned this last time around, I think, a high nucleus to cytoplasm ratio, which is something that helps control important characteristics of dynamics of the cell cycle, right, S phase, in particular that synthesis phase of the cell cycle in which we replicate DNA. And again, how do you take care of these PSCs, right? We've put a couple of basic guidelines at the bottom here, which I'm sure many of you are well aware of at this point, but we have to think about how cells are passaged, right? How frequently the media needs changing. We have to visually inspect the cells, right? We have to look for different features. We should remove differentiated cells if needed. We touched on that a little bit last time around. And again, it's always important, big picture, to plan accordingly, right? You should always prep your dishes. You should always maintain a sterile environment. You should also think about who's going to be working in the lab alongside you, right? You should also think about looking ahead to an extent, right? So you can anticipate and maybe prepare for the next steps in your respective workflow. One other thing to think about, and we did talk about this a little bit with feeder-dependent systems, right? They can nowadays quite easily be transferred to feeder-free systems as well. When we do this, it certainly works, right? You will see some residual feeders for a few passages anyway, but you can certainly make the switch from feeder-dependent to feeder-free culture quite easily nowadays. There's also a number of ways to visually inspect your cells. Again, we looked at confluency a few slides ago, but it's always nice to check in on things like confluency, things like morphology of your cells, right? You want to look for, in this case, things like clumping. You want to look for debris, changes in media color. Even with the naked eye can give you a good indication of how things are going at the cellular level. Most experiments will use or require a microscope, I would say, with at least a 5x objective, and I would say phase contrast capabilities are also ideal. A lot of folks, I'd say probably most folks nowadays, also use immunofluorescence microscopy, right, to observe things like morphology and identify specific markers. Again, most functional labs, I would say, would have these different tools on hand. And then lastly, when you're growing PSCs, yeah, go for it, Omar.
Just one quick comment about the cells.
Yeah,
Just
please.
generally speaking, I wanted folks to also know here that there is inherent differences between different cell lines that have different sources that you might see, right? So you might be working with multiple patient-derived cell lines. We talked about reprogramming last lecture. right? So you can picture, for example, reprogramming multiple different samples from different patients. And so you will see inherent changes in how these lines behave in culture because of the genetic background usually associated. Plus, there's honestly, some things that we still don't understand about those differences. Which again, to my point earlier about reducing variability as much as possible will help you better understand and optimize your cultures. But when it comes to the cells themselves, right, the kinetics of how they grow might be different. So different cell lines might be proliferating faster than others. Certain cell lines, you'll see even their morphology will be slightly different than others. There are acceptable thresholds, I would say, to Tom's point, how the morphology assessment is important, right? Because you want to keep track about of how these cells look, but also how they potentially change over time in culture, because that's not what they're supposed to do, right?
Absolutely. So passaging, right, is an important consideration. We also, when we're talking about passaging, there's a lot to consider even within that. We have to consider, say, the timing of passaging, how you're passaging, right? The timing of passaging is typically based on, from a big picture anyway, the characteristic growth pattern of cultured cells that I've put in this graph here. These are semi-logarithmic plots that show cell density versus time spent in culture over here on the right. We have this first phase here that typically occurs, speaking about the cells. And it's kind of a lag phase, if you will. It's this period of sort of slow and steady growth, and the cells are very much kind of adapting to their cell culture environment. But that lag phase that we see here is typically followed by the log phase, or the logarithmic phase. And this is, as you know, a period when the cells can actually proliferate quite rapidly. They consume a lot of the different nutrients in the growth medium. This is when we're using up all the media. And then when that growth medium is spent, or after a subsequent amount of time, right, when one of the nutrients is depleted, or when the cells occupy all the available substrate, the cells enter what's known as a stationary or plateau phase, in which proliferation is gently or greatly reduced, or sometimes even ceases entirely. And so there's kind of a sweet spot for passaging that I've marked here with an arrow on the graph, right? And that typically, I'd say in most modern workflows, amounts to about 80 to 90% confluency in adherent culture anyway. And again, what this does is keeps the cells at, say, an optimal density for continued growth. It allows them to stay happy, stay healthy, which further stimulates proliferation and basically maximizes the number of cells that you'll have available for your experiment. Again, just as an aside, because we will talk about this later, cell culturing suspension cells is often less complicated than passaging adherent cells, because the cells, as you think about it, right, in suspension culture, are already suspended in that growth medium. There's no need to detach them from the surface of, say, the culture vessel, oftentimes, again, making the process a bit faster and less traumatic for the cells when we're working in suspension culture. Just briefly, and I thought this would be a nice point of reference to have, I've listed a couple of tools here for passaging adherent cells. Things like gentle shaking, scraping, enzymatic dissociation. Obviously, all of these have their merits as far as different applications go, and the different dissociating reagents that you would employ along the lines for each. Again, I know we talked briefly on the notion of suspension culture, and we'll talk about passaging under those circumstances later on. And then lastly, switching gears here just one last time, there are just a couple of gentle reminders here about some of the challenges or hurdles that you're almost invariably going to encounter when you're working with cell culture. Again, I'm sure we all have some nightmare stories of things that have happened in the lab. We had one in my lab when I did my postdoc, a big mycoplasma infection. We were all out of the lab for about 3 weeks or so, which we thought was great as a postdoc. Initially, I thought it was great, but it turned out to add 3 weeks to everything and slow everything down. So it was an absolute nightmare. But the point here really is, when you're working with cell culture, always consider that things can go wrong, and they can go wrong quite quickly, right? Pay attention to yourselves. I'm trying to drive home today. Pay attention to your culture conditions. Pay attention to who's working in the lab around you. Some people have really good lab etiquette. Some people have awful lab etiquette. Stay away from those people if you can when you're working with your precious cells, right? Be conscious of safety, proper sterile techniques. And again, what this will do in the grand scheme of things is save you an awful lot of time and energy, as well as resources, right? Which cost a lot of money nowadays, by preventing unwanted problems. right? So it's important to remain aware when you're working in the lab, because quite quickly, as I said, this can quite rapidly change the outcome of your experiment and what you're actually able to do. Again, you can really, if it's done well, though, you really can leverage well-designed stem cell disease modeling workflows. I know we keep coming back to this slide, but putting in the time and the effort to sort of piece these components together in an orderly manner always works out better in the long run. You'll end up with great data that's reproducible, data that's going to get published and make an impact, all from being consistent, being conscious in the lab, and then being impactful in your work. And then last but not least here, I want to take just a couple of the principles, and this is just another 5-10 minutes or so that we've talked about so far, and apply them in the context of a case study. I picked this one today. It's short and sweet, but I think it's super interesting because what we looked at is cell line engineering to generate, as I said, hypoimmunogenic iPSCs, but this was actually work that was done in collaboration with a customer that we've had for quite a long time now, who's an absolutely wonderful person. If you've ever seen her talk, her name is Sonia Schrafer. She's at UC San Francisco. She's a physician scientist. She's super smart, super kind. And as I said, she's been a long-time collaborator with Thermo Fisher. What they did in this particular experiment is they generated a hypoimmunogenic derivative of iPSCs, right? One that, as the name implies, hypoimmunogenic, basically can evade immune rejection when it's transplanted into a recipient, in this case, an fully immunocompetent recipient. So what we know happens during pregnancy is that the maternal immune system is basically tolerant of different antigens, especially allogeneic paternal antigens early on, right? But what will often happen, usually transiently over time, is that they'll reject cells from, say, even the same baby later on in life. And so taking that kind of notion here, Dr. Schrafer looked at gene expression patterns of the syncytial trophoblast, right? The syncytial trophoblast is the cell type that we know is directly at that maternal fetal interface. And she postulated that those hypoimmunogenic iPSCs could actually be generated through mimicry of those different gene expression patterns, right? Those found at that syncytial trophoblast, at that placental barrier, right? More specifically, through an activation of MHC class 1 and class 2 target genes, and overexpression of CD47, which in this case they hypothesized would allow the cells to basically avoid recognition by the immune system. CD47 is super cool. It's basically something that acts as kind of this don't eat me signal, if you will. It prevents macrophages and other immune cells from basically engulfing and destroying healthy cells. And so what Dr. Schrafer did is took those iPSCs, she differentiated them into various cell types, so endothelial, smooth muscle, cardiomyocytes, and then transplanted those cells into recipient mice to basically assess their survival and immune interactions. So in line with a little bit of the content that we discussed today and in the first lecture and during Omar's, from a genome editing perspective, and again, we've got a whole lesson on genome editing, but a knockout here was performed in human and mouse iPSCs using a CRISPR-Cas9 system, looking at two specific genes, B2M and CT2A, which we know are associated with class 1 and class 2 MHCs. And we simultaneously targeted those for CRISPR-Cas9 mediated disruption. We then made a transgenic line of that double knockout with CD47, in this case using a lentiviral approach. So in other words, those edited iPSCs were transduced with a lentivirus carrying CD47, which ensured its overexpression. And then, as I mentioned, Dr. Schrafer's group basically differentiated those engineered iPSCs into cardiomyocytes and endothelial-like cells, right? But having done that, kind of the million-dollar question, as you might imagine, was this actually going to work, right? What was going to be the impact of this? And so I just pinched one figure from what's a really nice paper that was published in Nature Biotechnology in 2019 that essentially demonstrates that they were successful in generating hypoimmunogenic iPSCs, and that these cells could be differentiated into hypoimmunogenic cell types. In this case, what we're looking at here is the cell lines transduced to express a firefly luciferase reporter. Luciferase reporters are something that enables tracking by bioluminescence imaging, and showing here the transplant studies of those engineered grafts, again, in a humanized model here, a mouse model. So what we're seeing in this graph is a nice example of cardiomyocytes, right, where they differentiated both the wild type and the hypoimmunogenic iPSCs. You can see that based again off of that luciferase signal that they introduced into cells. Early on here, both the engineered iPSCs and the iPSC-derived cardiomyocytes quite nicely initially evaded immune rejection when they were grafted into that humanized mouse model. But if you look over here on the right of this graph, only the engineered cells survive long-term without the use of immunosuppression. While the wild-type iPSCs, right, those iPSC-derived cardiomyocytes, were cleared quite quickly by the immune system. You can see here that the wild-type cardiomyocytes disappear around day 20 or so, whereas the hyperimmunogenic cardiomyocytes maintain a strong signal for over 50 days. So again, just to take home here is that they perform gene editing. They used CRISPR-Cas9 to do so, and a lentiviral-mediated approach. They generated these hyperimmunogenic iPSCs. They differentiated those into terminally differentiated cell types. And because I would say those engineered iPSCs, as I showed you in that last graph, really evaded immune rejection and actually survived for quite a long period, at least 50 days or so after engraftment. Again, I think this speaks to the potential in the realm, in this case, of, say, transplantation therapy, and was a nice story. And the papers, as I said, is really interesting as well. Along those lines here, I'll put a link for the paper in the resources that we share for today, along with an outline of some of the content we talked about and a few other useful resources as well. But I'm going to stop there in the interest of time. Again, thank you guys very, very much for joining us. Hopefully this was interesting, maybe even a bit useful. Just a fresh reminder of some of the tips and tricks along the stem cell workflow. Omar and I'd be happy to take any questions, either now, offline. We can talk about this content today. We can talk about previous content or anything else you guys fancy talking about as well. But thank you, everyone, for joining us.
Thank you, Tom. Let's see, maybe while folks, if they want to drop some questions in the chat, if they have any. But while they do that, I will steal the ball for just a second, Tom.
Please do, yeah.
I wanted to share a resource that we can direct folks to that might be helpful here. If I can, I think it's this. Okay. So, can you see my screen okay?
Yes.
Excellent. Just for folks to know, we do have this link over here that I can drop in the chat, and then we can or we can share with the resources afterwards as well. But it's a bunch of how-to videos around some of the stuff that we're going to be talking about for the entirety of the lecture series. And so there's some really, really helpful videos, for example, on some of the topics we talked about today about selecting a matrix that's appropriate for your culture, right? Tom talked about the three different matrices that we use for feeder-free culture. This is, for example, the truncated vitronectin. But one thing I wanted to show is that we have, a little bit more direction on, what you want to dilute this in and how to store these things, and for how long, right? But there is another video that is also related to the passaging, different passaging reagents that you can use for different dissociation strategies with your cells, right? That Tom mentioned is an important consideration during your culture. And so do we have any, before I play this, let's see if we have any questions. Possible to use KSR and be mercapto feeder-free conditions, or does it only work with meths? So you can use KSR supplemented media for feeder-free cultures, but it tends to be quite difficult to maintain these cultures going in that feeder-free condition with just the KSR or FBS supplemented media. Okay, sorry about that. So the passaging video. Tom mentioned that, right? This is definitely one of the important considerations as the cells especially in 2D culture start to fill up that culture vessel, right? We start to become confluent. Usually around 75 to 80% is when we recommend passaging those cultures, which, as Tom mentioned again, just means basically taking those cells, basically cutting them up, if you will, in some sort of way, and transferring them over and diluting them over into another set of cultureware. And in this case, the reagent that you use will actually digest your PSC colonies differently, right? We typically categorize the different reagents by their strength. And this is a really, really nice video just to kind of give you an idea about how the just something as simple as your passaging reagent that you use can affect your cultures quite heavily, right? Fast forward to here. You'll see typically in the stem cell arena, and maybe I'll make this bigger. Is that better? Is that first full screen now? Excellent. Typically, you'll see one of three dissociation reagents that are commonly used. The first is versine. This is an EDTA-based chelating agent that is supposed to break cell junctions. It's what we call a gentle dissociation reagent. There is our Stempro acutase that's slightly stronger, can break the cells down, our PSCs down into a 1 to 2 cell cluster. And then you have the triple select, which is even a little bit stronger than the acutase, where you can truly go down to a single cell suspension. But if you remember, Tom mentioned that stem cells do not like to be on their own, right? So the degree at which you separate them from each other can cause them varying degrees of stress, which is why you really have to be careful what dissociation reagent you use during routine culture or for specific applications. And for example, if you're using the versine solution, you see there's this time-lapse video of this colony. You'll start to see, those like little holes after a couple of minutes as these cells dissociate. And then simply pipetting them up and down can create these smaller clumps that we then recommend passaging over during routine culture. So this is how I would passage normally. Once these clumps attach, you'll see that they grow and then they maintain that really nice PSC morphology 24 hours later, right? And this is done on Geltrex matrix with StemFlex medium. I mentioned the Stempro acutase is a little bit stronger, right? This is more of a proteolytic enzyme. And you'll see that right away that the dissociation strength of this reagent is definitely higher than that of versine, and that you can go down to those 1 or 2 cell clusters quite easily. Now, when you take those 1 or 2 cell clusters and you transfer them over, I want you to notice how the cells, once they attach 24 hours later, which is right now, they look a little bit stressed out, right? So they still have a somewhat of a tight, compact morphology, but they're a little bit more spindly looking. And that's because the cells were digested down into that 1 to 2 cell cluster, right? Making them a little bit more stressed, but they typically recover in appropriate conditions after a while. And then finally, triple, which is the recombinant enzyme to really dissociate the cells down into single cells. Again, you'll see very, very clearly here that once you add the reagent, you'll start to see those individual cells really be digested down to that level. And when you plate those single cells over into a new plate, again, on Geltrex you'll see that the morphology is going to be even more spindle-like, or more, stress fibers are going to be very visible because, again, PSCs do not like to be singularized on their own. Again, under the right conditions and the right media, the cells can recover, especially if supplements like the Revitacell or RUC inhibitors are added. But if you routinely culture these or passage these cells and dissociate them down to single cells, then you're going to eventually cause some sort of drift in your culture, right? Which is why we typically recommend, as Tom mentioned, for routine culture, we typically recommend clump passaging with something like the versine reagent as well. So again, big rant. Sorry, I warned you guys that I talked too much, but this is a good resource. It's a really nice set of videos. There's some lectures in here as well. Videos on how to, cryopreserve cells, bring cells out of thaw, and we'll talk more about the suspension-based culture system in the next lecture, and some of those videos are on there as well. I absolutely can share the link. I'll drop it right now, and we'll add it to the list of resources that we send as well too. Okay, we have one more question. Which one is better, triple select 10x versus triple select express? Um, Good question, Naresh, for beating cardiomyocyte or harvesting mark in a 2D in a 2D monolayer setting. If you just have 2D cardiomyocytes that you're harvesting, I would say triple select the 1x would probably be your best reagent. The 10x might be more appropriate for cardiomyocyte organoids because they become quite lumpy and deposit quite a bit of extracellular matrix. Dissociating that becomes a little bit harder. So using the more concentrated triple in that instance then really helps out, I think.
Yeah,
Excellent.
before wrap it up, and just here's a quick review of the lecture 4. And I'm not sure I can do it, but just for people interest. And another feeder-free human pluripotent stem cell culture advance. And the main issue of the in actual experiment is in the human pluripotent stem cell culture is clonal variability. So that is the really bothersome in actual your practical experimental design. And in this aspect, culture media condition is really important to minimize this clonal variability. So that is the reason why someone just asked earlier version of the culture media condition versus nowadays culture media condition, huge difference. And the clonal variability is really, really minimized nowadays. What does that mean? Just learning the history of the media development and also various agent development with just a rationale behind. This knowledge will be really invaluable for troubleshooting and then optimizing your pluripotent stem cell experimental design. And actually that is the reason next lecture will provide with practical tip and the foundational insight. So therefore, I really eager to attend the next lectures. And hopefully everyone join the next lecture.
Yeah, I think it's going to be super exciting because we can talk exactly about that to Kitai, where mention a little bit more about, even some of those processes that are related to clonal expansion, specifically in the context of PSCs. Like you said, we're trying to reduce variability. When you're disease modeling, you want to work with one genotype. You want to make sure that you have a clonal population. What are the considerations there? What do you need to think about? So yeah, I'm excited. And we'll talk about suspension-based culture as well, which is super, super cool. We get to show some of that too. So that'll be really exciting.
Hey, Omar, Tom, could I ask a question quickly before we log off?
Absolutely.
Hey, this is Tyler. Really appreciate the insight. The links are great. I guess one thing, just to kind of elaborate on my prior question, we've talked about passaging based on confluency, and you showed some nice images of healthy iPS colonies. I guess in the context of, initially generating your iPS clones, so you take a primary human sample, in our case, we do Sendai reprogramming. And then over time, we want to select out individual clones. And this is also applicable for when we're doing subcloning, to really make sure that we have individual clones. We'll see these nice colonies start to grow out, and then they have this really nice morphology. At least in my hands, if we let that go too long, they start to clump up in the middle. We get some differentiation in the middle and potentially the edges as well. So, we talked about techniques for passaging those with needles or the dissociation reagents. But when do you kind of make the call to passage a colony, when it's got that nice flat morphology before it starts to get heaped up in the middle and differentiated on the edges? When do you guys kind of make that call?
Yeah, that's a good call up because it would depend not just on confluency, Tyler. If you have, for example, especially during that initial stage where you're setting up a clone where you may have just one or two, colonies in a plate, having that grow out and fill up the entire surface area is not optimal because you're going to eventually, run into the problem that you're mentioning where the cells in the middle start to clump up or differentiate. On that front, usually I don't like to have my colonies go above a the 500 micron mark, like and yeah, kind of like the below 1 millimeter, if you will,
Mm.
diameter across. And if it's above that, then you have to really watch the edges of the colony and particularly the center as well. There has been times where if it's a precious enough sample, and I want the cells to proliferate a little bit more to get slightly more cells in the process before I passage, I will carve out the center of the colony with a sterile tip. But again, to Tom's
Mm.
point, just you have to make sure that you're being very careful that the more handling and manipulation, the higher the contamination risk is.
Yeah, yeah, appreciate it.
Hi, Omar. Do you have time to take one more question?
Absolutely, yeah, absolutely.
Yeah, yeah, it is a very wonderful session today I have. My question is related with like thawing of cardiomyocyte. So I have differentiated iPSCs to the cardiomyocyte, and after enrichment, I found that those cardiomyocytes were more than 90% troponin T positive.
Nice.
Then I did freezing of those cardiomyocytes. Then I wanted to do thawing. So for thawing, I have used cellular dynamic plating media that
Hmm.
comes with their kit. So because I do not have the proper conditioning media that was planned for my experiment.
I see.
So when I use that cellular dynamics plating media, the viability of the cardiomyocyte like very, very poor. Maybe like 10% of the attachment was there. Like
Mhm.
90% of the floater were there. And
Yeah,
those attached looks like those are fibroblast contamination, I guess.
contaminated.
Uh,
Yeah, you
yeah.
might you might
So,
still see some like mesoderm formation, even though you have a high troponin 2 cells, you might still see some fibroblasts. But
mhm.
typically after dissociation, these cells are quite fragile, so getting them to stick back down and attach is quite difficult. Naresh, if you email me, I can send you a protocol that we've been working on for replating of cardiomyocytes. If that's of interest to you, I can share it with you as well. I don't think there's
Okay,
anything special, but we add a little bit of DNase and things like that that I think might be helpful.
okay, great, Thomas. How can I get your email? Is there
I can
any?
drop it in the chat. Yeah, I can drop it in the
Okay,
chat for sure.
okay, great. Okay, thanks so much. Yeah.
Of course.
So we have include lecture for cardiomyocyte, right? Just I want to confirm. Okay,
Yep,
great.
yep. And we have
Absolutely.
some internal data on it as well that we can share too.
Yeah, it's scheduled in week 7.
Yeah.
Yes. And I will, just for folks, I'll write my email down. I know there's already Emma that reached out to me. All right, I'll wait for your email as well. But if you guys need anything, you definitely can reach out to myself or Tom. We're definitely happy to
Anytime.
help. And another quick shameless plug too, but for most folks, especially on the UCLA side, um, use your resources well, right? So the core is I can't explain how much of an invaluable resource this is. To be completely honest, it's doing this kind of work completely on your own from start to finish is not the simplest thing in the world. So don't you don't have to reinvent the wheel. There's a lot of expertise, and I speak for myself and Tom, but I'm sure on Kita and Catherine's side too that they're more than happy to help out with any requests you may have. So if you have any
Absolutely.
questions, reach out. We're happy to help. Awesome. Anything else on your end, Tom?
No, that was it.
No. And thank you for that. Was a nice lecture today.
Absolutely. Yeah, no, thank you guys. Hope you all have a really, really good week, and we'll look forward to seeing you next Monday.
Yeah, see you next
Perfect.
week.
Thanks,
All right.
everybody. Have a good
Appreciate
one.
it. Bye-bye.